Hi, my name is Alexandre Coudray. I'm a PhD student in Didier Trono Research Group. And today we'll summarize you the ongoing project we have with the Swiss Data Science Center. So we're trying to predict the drug sensitivity at cell type resolution using a machine learning framework. In particular, we're interested in predicting drugs with higher efficiency to target specific subpopulation of cancer cells. So if one wants to do that experimentally, you will need to use purification techniques and then to pass the sorted cells through a drug sensitivity screening. However, this requires typically a large amount of cells as input and leads only to imperfect isolation of cell types. Also, it can impair the viability of cells and thus bias the drug sensitivity screening readout. So the solution we propose is to demix drug sensitivity of cell population through a machine learning framework, and therefore we can avoid any purification step and reduce the costs, and maybe also improve the accuracy. So the experimental design involves two main steps. The first one is to infer cell type proportion through a so-called Bold deconvolution method. So we're trying to push the field here and to improve existing Bold deconvolution methods. The idea of Bold deconvolution is to use gene expression to infer cell type proportion and typically rely on some single cell RNA sequencing data. Actually, the main novelty we bring is the second methods, which allows us to integrate cell type proportion previously inferred with drug sensitivity signal from the mixtures and to deconvolve drug sensitivity at cell type resolution. This is done by considering latent variables contribution to the mixed signal, and we resolve it through an expectation maximization algorithm that we were able to derive thanks to the contribution of the SDSC data scientists involved. Thus, what we are doing is to model the probability to survive a treatment of each cell type and their contribution to the observed survival rate at the bulk level. Ultimately, we get a prediction at the cell type level by integrating information from multiple samples. To believe this work might have a great impact in the field, we envision those computational methods could replace costly experiments necessary to screen drug sensitivity at the cell type resolution, which can be long, tedious, and expensive. Also, in the specific context of acute myeloid leukemia, we are able to predict drug sensitivity of leukemic stem cells, which is of high clinical interest. Thank you very much for your attention.